So now let's go over all of the parts that make up a cell, aka the organelles. So the first one is a cell wall. This tends to be on the outside of the cell. And during our macromolecules lecture, we talked about polysaccharides and how the cell wall is usually made up of polysaccharides. So cellulose is a component of a plant cell wall. Peptidoglycan is a component of a bacteria cell wall. And chitin is a component of a fungi cell wall. Okay? So this cell wall usually gives that cell support and makes it hard. Okay, so it's like an outer hard covering. And so now which cells have it? So plants, bacteria, fungi, not animal cells. Then underneath of that cell wall, you would find a cell membrane or a plasma membrane, or as the outer covering of an animal cell for our intent and purposes would be this plasma membrane. Again, from the previous macromolecules, you should remember that this is made up of a phospholipid bilayer. So we have that polar head and then the nonpolar tail molecules, right? And so this phospholipid bilayer made up of lipids peppered with proteins so they can form channels, or they can also be on one side or the other. All cells, okay, so bacteria, plants, animals, fungi, they all have this plasma membrane. So what does it do? It serves as like the gatekeeper, allowing only some things to enter and keeping other things out, okay? And how those things enter depends on what kind of molecule it is. It could be carried across by a protein. It can go through a protein channel, or it may be able to just squeeze on through that lipid bilayer, okay? Then we would have this cytoskeleton. And so this runs underneath of that plasma membrane, and it's going to give structure to the cell. So you'll find these in plant and animal cells. Now, interesting to note here, so I'm sure that we have all heard of sickle cell anemia. Well, sickle cell anemia is caused by a mutation in a cytoskeleton component. And so by mutating one component of the cytoskeleton, you go from having a donut-shaped red blood cell to this sickle-shaped red blood cell. Okay, so the cytoskeleton, although we don't think about it much, is very important to our cell. Then you have the nucleus. This is that brain of the cell. Okay, this is where you're going to find the DNA that's protected within the nucleus. It's going to control the activities of the cell. So the nucleus really is analogous to the brain of a human. You find this in plants and animals. So here's our nucleus. They have a pore that's also selective about what comes in and what goes out. You'll find the DNA. There's a nucleolus here, like a brain within the brain, all right? And then you'll see as we go through that extensions of the nuclear membrane makes the rough endoplasmic reticulum. Now, the rough endoplasmic reticulum has ribosomes on them. Those ribosomes are made up of two components, a small and a large subunit, that interact together to make the proteins. You'll find this in our plants, our animals, and our bacteria. That rough endoplasmic reticulum I was talking about, an extension of the nuclear membrane, peppered with ribosomes. It's called rough because when looking at it, the ribosomes give it this rough appearance. So its job is to help modify and transport proteins. So remember that DNA has to be converted into mRNA to help make our proteins. Well, we have those ribosomes that are on the rough yard capable of making proteins. After it makes it, we can use this endoplasmic reticulum to modify and transport those proteins where they need to go. We're going to find this in plants and animals. Then we have that smooth. So keep going from the rough, you'll get to the smooth ER. Characteristic is it doesn't have that bumpily appearance like the rough ER because it doesn't have ribosomes. What happens here, among other things, depending on the type of cell, though, is that it makes lipids, okay? And the same place you find the rough is where you find the smooth in your plants and your animals. And we have the Goji apparatus, also called the Goji body. This is the classic quote-unquote post office of the cell, okay? So you're going to go to the Goji, it receives a package, opens it up, takes the contents out, modifies it, repackages it, and ships it where it needs to go. This is in your plants and animals. I mean, this is important because if you look at the cell, and know that, hey, if I make a protein in the ER that has to get to the cell membrane, how does it do it? Well, that's where the Goji comes in and is able to put those things where they need to be. And not just proteins, but any molecules that can be made within the cell can travel through our Goji system. The lysosomes have budded off from Goji, and their job, they're filled with enzymes and things inside that help them to break down. These are like the recycling center of the cell. So they'll break down those damaged macromolecules that you should be able to name, and they'll break down the cell parts that we are talking about. So if there's a damaged ribosome or something, lysosome comes along and says, oh, you're out of commission, let me break you apart, and then possibly those parts can be recycled to be used again. And you'll find this in animal cells. And then peroxisomes are also these small little molecules. And in the inside of them, they have this urate oxidase, a type of enzyme. And its role is to help detoxify our cell, okay? It can break down long-chain fatty acids and other things that the cell no longer needs. It's outside, like most of them, have this lipid bilayer. So remember, these are membrane-bound organelles. You'll find this in your plants and your animals. We have our chloroplast. We should all know what the chloroplast does. And so this chloroplast is very interesting, has a double membrane, has some ribosomes of its own, even has some DNA of its own, okay? Really cool organelle. It's green. It has chlorophyll. It gives plants the color green from its chlorophyll. It's involved in photosynthesis. What is photosynthesis? I'm hoping that you're thinking that, hey, this is how we convert light energy from the sun into glucose, okay? This is also where we can give off, through photosynthesis, we give off oxygen. You find this in plant cells. Powerhouse of the cell, aka the mitochondria. I know we all heard about the powerhouse of the cell, but what does that mean? Okay, what is the powerhouse of the cell doing? It's making cellular energy, so this is ATP, adenosine triphosphate. This is involved in cellular respiration. You can think of this as the yang to chloroplast, okay? So this would be almost the opposite. It will take in that glucose and then give off some other things. And you'll find these in plants and animals. So plants have chloroplast and mitochondria. Animals have mitochondria. Similar to chloroplast, again, it has its own DNA and ribosomes. It has a double membrane. Then we have a couple organelles that help the cell move. And so when you first seen the bacteria cell, I pointed out cilia and flagella. So the cilia beats back and forth like an oar, like back, forth, okay? And you'll find this on the outside of some bacteria and some animal cells. So for instance, our respiratory tract and the reproductive tract of females are lined with these cilia that help things move, okay? So in your respiratory tract, you can't have dust and things hanging out. So it's constantly flapping the cilia to keep this pace going, to keep your tracts clean. Then you have this flagella that moves like a whip. Crack the whip, okay? And now you'll find this on some bacteria helping them get around. But you'll also find it on the sex cells of some plants and animals, okay? So that also helps them get around. Classic example will be sperm in male animals. They have a flagella that helps them to swim. Then we'll go back to the cytoplasm. So remember, this is one of the 4 parts that all cells have. This is like a jelly-like fluid that you find in the inside of a cell. So this could probably be analogous to like the air in your home, the space, what fills the room, right? And so here it's a lot of things going on, even though it doesn't seem like it. So it can help organelles move, so it has like this streaming, but it's also the site of all of the, not all, but a lot of the metabolic processes of a cell take place in this cytoplasm. We have the vacuole. So if we think back to that plant cell, we saw this large centralized vacuole. This holds storage, okay? Depending on the type of plant, it can hold different types of things. It's found in plants. Now, it can also be found in animals, but it's very small because it doesn't have the same things that it doesn't do the same things that a plant needs it to do. Another thing it helps plants with is turgor pressure. Animals don't have to worry about that. Now, there are some animal cells, depending on the type of cell, that can have a large vacuole. For instance, fat cells, okay, adipocytes. They hold adipose, aka fat, so they tend to have these large vacuoles because their job is to store that fat. And then we have centrioles. So our centrioles help us with mitosis, and we'll talk about that in the next module. And so they go to the ends of the cells and help us to organize our chromosomes so we can pull them apart during mitosis. And so they usually have this 3 to 9 arrangement, so 3 that are bound together and then 9 in the circle, and they tend to be at this 90 degrees to each other when they are working. And you'll find these in animal cells. Plant cells are capable of mitosis, but they don't use centrioles to accomplish that. And now this one is missing from your list, so you'll need to write down proteasome. These help us to degrade damaged or unnecessary proteins. So these are like lysosomes just for proteins. They're going to help us go in, break them down, and then reuse those proteins. We have a system in our bodies called ubiquitin or ubiquitination, and what it does is put these ubiquitin tags on proteins to mark them for degradation. So think of it like spray painting. Hey, this protein needs to be degraded. And you'll find this in plants and animals. Okay, so we've talked about all the organelles and cell parts that you find in plants, animals, and bacteria cells, and to a lesser degree, I've mentioned some that you find in fungi. Now, I've also mentioned the characteristics that you find in chloroplast and mitochondria. And the reason being is that they're tied to how we think organelles originated to begin with. So remember, and when we talk about evolutionary history, we go from simple to more complex. And out of prokaryotes and eukaryotes, which one is simpler? Those prokaryotes, right? So you see this prokaryote and how it doesn't have all the stuff you find in the eukaryotes. So we think that that evolved first, and then over time, a prokaryote got really fancy and evolved into what we now know as a eukaryote. So what fanciness happened? So there's this endosymbiotic theory that was put forth that has lots of evidence to support it that says, hey, this is how we have organelles. And the first thing is one cell lived inside of another. It's hypothesized and supported that mitochondria and chloroplasts used to be bacteria cells. That's why they still have ribosomes and DNA, because they once were free living cells. And then they lived inside of another to help them, like, hey, the ancient chloroplast was able to photosynthesis. Another cell might have needed that photosynthesis, so it engulfed it, and then they worked together to survive and to thrive in whatever their environment was. And so this is the endosymbiotic theory. And then we know that a lot of our organelles have the same plasma membrane components, that lipid bilayer. So we think that there was some enfolding that helped to form them. And then, evidence to support is that they're made of the same thing, as well as the connections between the nucleus to the endoplasmic reticulum and things like that. Okay, so any questions, comments, or concerns, write them down, bring them to class, and we can talk about it.